I will be brief in introducing our next very accomplished, very well-respected speaker. He is Dr. Frank LaFerla. He's a professor of neurobiology and behavior and co-director of UCI's Institute for Brain Aging and Dementia. Really a man at the forefront of Alzheimer's research, made many key discoveries. In fact, he was the first to engineer mice that developed the plaques and tangles that characterize Alzheimer's disease. And that provided this living lab that not only informed his work, but it was so important to research around the world. He has trained as a microbiologist at the University of Minnesota, and in his postdoc training, he became interested in this whole notion of putting human genes into the mice. And so he transferred to Red Cross Labs in Rockland, Maryland, which was one of the few laboratories doing that kind of pioneering research at the time. And the LaFerla Lab is focused on understanding the molecular mechanisms leading to degenerative diseases of the brain. We have just met, and I am so glad that we now know each other, and now I understand what all the fuss is about with Dr. Frank LaFerla. Welcome.
Thank you.
To your own home, I should say. You're welcoming us.
Yes. Thanks. It's a great honor to be here today and to have this opportunity to talk to you about some of the new work that's being done in my laboratory. What I'm going to talk to you today is about work that has not yet been published, and it's still under investigation. Okay, thanks. I'll do my best. So let's just review a little basic facts about Alzheimer's disease. We've heard some of them already. We know, for example, that Alzheimer's is an age-related progressive disorder that generally destroys a person's recent memories. That recent memories are usually the first cognitive domain to go, but the patients undergo a lot of other changes as well, including experiencing some psychiatric problems. They have difficulty with their judgments and many of the other things that we've heard today. The time from initial diagnosis to death varies pretty markedly from individual to individual. It could be as short as 3 years from the time of initial diagnosis, or sometimes as long as 10 years. The average, though, is about 6 to 8 years. So, why is this such an important disease? Well, just based on the numbers alone, besides what it does to us medically. Right now, there are 5.2 million Americans that are afflicted, over 20 million people worldwide. Age is the most significant risk factor for Alzheimer's disease, and 5% of the population over the age of 65 is afflicted with this disorder, and that number doubles every 5 years thereafter. So think about that. We're talking about 5% over 65, 10% over the age of 70, 20% over the age of 75, and 40 to 50% of the population over the age of 85 is afflicted with Alzheimer's disease. Right now, we consider it the 6th leading cause of death in the United States and the 6th most expensive illness to manage. However, depending upon how you do the math, it could actually be bumped up into the 3rd leading cause of death and the 3rd most expensive, because the other disorders that are in front of it are different forms of cancers. So if you lump the cancers and the coronary heart disease into a single category, then Alzheimer's disease becomes the 3rd most expensive disease and the 3rd leading cause of death. And it's a problem that's going to get worse progressively over the years, because we as a society have had great success in elongating human lifespan. And it turns out that age is the most significant risk factor for Alzheimer's disease. And so here we have about 5.1 million people that are afflicted today. And if the trend is not altered, by the middle of the century, we're talking about 16 million people that are going to be impacted with Alzheimer's disease. And so this is going to have tremendous impact for this country, but also for the state of California, in part because over 10% of the nation's Alzheimer's patients live in the state of California. There are over 588,000 people living in California with Alzheimer's disease. In Orange County alone, there are about 40,000 individuals afflicted with Alzheimer's disease. What's worse is that by 2030, which is only 20 years away, the number of Alzheimer's patients in this state will more than double to about 1.1 million people. And because of the demographics, the number of Latinos and Asian Americans living with this disease will triple, and the number of African Americans will more than double just in the state of California alone. So at present, we're spending about $53 billion just to care for Alzheimer's patients. So there is clearly an urgent need for us, particularly at the state level, to focus on Alzheimer's disease, in part because our long-term care costs in the state of California are much higher than what they are nationally. So you could see that from 2004 to 2008, our long-term care costs were growing at a rate of about 44% compared to the national average of about 17%. So what is going on in the Alzheimer's disease brain? Well, actually, Dr. Alzheimer's himself in 1907 described some of the characteristic changes that occur in this disease. And simplistically, we consider this disorder to contain two pathological hallmarks, what we refer to as plaques and tangles. And the plaques look like these Brillo pad structures that get deposited in specific areas of the brain that are related to learning and memory. And generally, these structures occur outside of the cell. And they consist of a small peptide, which is called beta amyloid. Now, Alzheimer's made a mistake in the early 1900s by naming these amyloid plaques because amyloid implied that they contained starch. And we know that they do not contain starch. They actually contain this very small peptide called beta amyloid. The other structure that he identified over 100 years ago actually occurs inside neurons, and these are referred to as neurofibrillary tangles, and Rod mentioned those during his talk. So these two lesions are required for a diagnosis of Alzheimer's disease. So where did our lab come in? Well, my lab had developed the first mouse model that recapitulates both plaques and tangles. And I'll save you, for the sake of time, all the details here, but essentially what we did was we made a transgenic mouse from a transgenic mouse. And the bottom line is that these mice develop both plaques, which are shown here in green, and neurofibrillary tangles, which are shown here in red. And so this has been a significant advance for the field. As a matter of fact, we've actually sent these mice to over 120 investigators in 20-plus countries throughout the world. And still, even though it's been about 4 or 5 years since the initial description, get on average about 12 emails a month requesting these mice. So we have used these mice to investigate a lot of therapeutic treatments, and we're happy that many of the compounds that we've evaluated in the mice have made their way to clinical trials. However, I want to point out that not every treatment that we investigate in the mouse model actually has a positive effect. As a matter of fact, most of what we test fails. However, as scientists, we generally tend not to report what has not worked. And that has had a very adverse effect because sometimes people have this assumption that everything cures Alzheimer's disease in mice, and that is not true. And so these are just a listing of some of the things that we have tried in our lab that have either failed to improve the pathology or to improve the cognitive phenotype. Now, what you'll notice from this list is that there's a large bias, and I think indeed the field itself is largely biased towards small molecule therapies for the treatment of Alzheimer's disease and other neurodegenerative diseases, and have largely ignored cell-based therapies like stem cells. And so I have to admit up front that I was one of those people who was very skeptical about whether or not stem cells might actually play a role in Alzheimer's disease. And so we decided to actually let the science answer the question for us. And so our question was very simple. Can neural stem cells affect AD-related pathology and cognitive decline in our animal model? And to do this, what we did was we started off with mice that were 18 months of age. These are very advanced aged animals, and takes a long time to get to 18 months of age. You have to wait for them to age. You can't go out and buy them. And it means that our mouse bill, our hotel bill for these mice is very high. We pay about $25,000 a year, $25,000 a month just to house these animals. And that was meant for Sue's benefit. So we started off with these animals that were 18 months of age. By this point, they have very severe cognitive decline, and they have very advanced pathology. You can see here's the plaque pathology, here's the tangle, here's the merging. And what we decided to do was inject these animals bilaterally into the hippocampus, as this is one of the main areas that's afflicted in Alzheimer's disease and in our mouse model. and wait for about a month's time, then reevaluate the mice cognitively, and then sacrifice to see what was going on. So the cells that we put in, we were lucky. They were expressing a fluorescent marker. And so that meant it was very easy for us to track them after we implanted them into the brains of our animals. And these cells, these neurostem cells, were derived from a transgenic mouse, as I said, that expressed this fluorescent marker. They were from P1 stage. And what we found was that these cells were able to express all the markers of stem cells and to show that they were multipotent. So that was a little nice control. In addition, we showed that these cells were able to differentiate into all three neuronal lineages. So they could differentiate into neurons, they could differentiate into astrocytes, and they could also differentiate into oligodendrocytes, even though that's not shown here. So we were very happy about that, and now wanted to know what effect does this transplantation have on the cognitive phenotype in the mice. So how do you measure cognition in mice? Well, an easy way to do this is using what's known as the Morse water maze. This is just a simple circular pool, and you can see that there's a platform here. And the first time you put in a mouse, it's unexperienced with this environment. It just swims around until it randomly happens to bump into that platform. If it doesn't find the platform within 60 seconds, we just physically pick it up and put it onto the platform just to let it know that there is an escape route. And after a few days of training, these mice actually learned this task very well. And normal mice, after that time point, are able to find the platform pretty quickly. And you can actually measure the time it takes to find the platform as an indication of how good its memory is. So we use this task to determine whether or not the stem cells were able to improve the cognitive function in this mouse model. And I know this is a complicated slide, but if we just focus here on panel F, what we can see here is what we were measuring is the latency. That is, how long does it take for these animals to find the platform? And you could see the lower the bars means the shorter the time, and that's good. And the two left TM bars here show how normal animals are performing. Here in blue, you could see this is what the Alzheimer's mice were performing that did not receive the stem cells. However, if you look on the right-hand side in green, you can see these are the Alzheimer's mice that now receive the neural stem cell transplants, and now they're performing as well as the non-transgenic animals. And again, these are 18-month-old animals with very severe pathology. So we can show the improvement in cognition using a couple different tasks. One was the Morse water maze, and we also used another task such as novel object recognition. And so that was a very exciting result, and as always, it raises new questions, and that was, how does this transplantation improve cognition? And so we looked at a few obvious things. When we implanted these cells into the hippocampus of these animals, we actually implanted them right about here into the molecular layer, and what we found was that the cells were actually migrating to the dentate gyrus area, which represents a niche in the brain where they might be comfortable. But surprisingly, very few of the stem cells were actually migrating to the amyloid plaques. So it didn't seem that they were affecting the pathology by removing the amyloid deposits in the brains of these animals. Likewise, it turns out that very few of these cells were actually becoming neurons. In fact, the majority of them were becoming oligodendrocytes or astrocytes, such as what's shown on the top panel. Only about 5% of the cells that we transplanted into the brains of these animals were in fact becoming neurons. So we didn't think that stem cell transplantation was improving the phenotype by a neuronal replacement model. So that still left the question, how was this working? We know it wasn't by becoming new neurons. And so as Alzheimerologists, what we did next was to look at the amyloid and plaque pathology that occurred in these animal models. And what we found was that there was absolutely no difference. There was no reduction at all in the amyloid levels, whether we look and count plaques or actually measure it quantitatively by ELISAs looking at the different forms, even collaborating with Charlie Glabe here at Irvine to measure different oligomeric forms. This is unparalleled. This is the first time in our lab, and to our knowledge, that we have been able to improve cognition in a mouse model of Alzheimer's disease without lowering plaque pathology. Likewise, we had no effect on tangle pathology. And so again, that left a question, how is it that these stem cells are improving cognition if it's not by altering A beta or tau, which we thought was going to be the most obvious route? Well, it turns out that the stem cells were promoting synaptogenesis in the hippocampus. This was particularly interesting because if you look at humans who suffer from Alzheimer's disease, the best correlate of their cognitive decline is not the number of plaques and tangles that they have in their brain, but rather the degree of synaptic loss that they have. And here it turns out that by implanting these stem cells into the brains of these animals, here's what the vehicle-injected animals have, and here's what the animals receiving the stem cells. You can see that there's almost a doubling in the synaptic density following this. And this was particularly exciting to us. And it turns out, we tried to get that at the mechanism, it looked like these stem cells were actually secreting neurotrophic factors, and one in particular was of interest to us. It turns out to be brain-derived neurotrophic factor, BDNF, and you could see here's what the controls look like, here's what the neurostem cells look like. They have a marked increase in BDNF expression. And when we put them into the brains of these animals, it induces the surrounding tissue to also secrete more BDNF. And here's some of that data. Here's what the levels of the total levels of BDNF are in the brain. Here's what happens when you inject the stem cells. You can see that if you actually measure what cells are expressing BDNF, the green represent the cells that we implanted into the brains of the animals. Red represent the cells that are expressing BDNF. And now you can see that not all of the cells that we transplanted, it's not only the cells that we transplanted that are expressing BDNF, but the stem cells themselves induced other neighboring cells to secrete BDNF. And so that was particularly exciting to us. It then raised the question, can BDNF mimic this effect itself, or would neurostem cells be better? And so what we did was we actually injected mice once with BDNF, and what we found was that that was able to rescue the cognitive deficit in these animals as well, and likewise led to almost a doubling of the synaptic density in these animals over a short period of time. And reviewers are never satisfied, and they always want you to go further. And so what we next had to do was determine what effect does reducing BDNF in these neurostem cells have. And so what we did was we took a siRNA approach, which essentially means we could take these neurostem cells and eliminate expression of BDNF only in those neurostem cells. And here's the evidence for that. You can see we can reduce BDNF levels in the cells by about 78%. And now, when we implant these cells into the brains of our animals, we are no longer able to rescue the cognitive phenotype like we had earlier. So here's what the vehicles look like. Here's what happens when we implant the normal stem cells. You can see you get a very nice reduction in cognitive improvement here. And here's what happens when you reduce BDNF and implant them here. There's no longer statistical significant difference here. Likewise, when we measure synaptic levels in these animals, turns out that you get a very nice doubling with in the animals in which you implant this neural stem cells. But if you implant these modified stem cells that no longer secrete a lot of BDNF, you get a marked diminution of the amount of synaptogenesis. It's not complete because we only reduce BDNF levels by 78%. There's still some BDNF that's being secreted in these cells. So, in summary, it turns out that by transplanting neurostem cells, we're able to rescue hippocampal-dependent learning and memory deficits in very aged animals that harbor extensive plaque and tangle pathology, as well as other components of AD pathology, such as neuroinflammation, dendritic loss, neuronal loss as well. It turns out that these stem cells don't appear to be improving cognition by affecting neuronal differentiation or by altering A beta and tau pathology. And for us, that's a very novel finding. Rather, it seems that its mechanism is related to BDNF and by increasing synaptogenesis. And BDNF for us seems to be an important developmental candidate because we can show that by injecting BDNF into the brains of these animals, we can mimic some of the effects, not all of them, the stem cells are still better, in terms of improving the cognitive phenotype. And likewise, if we remove BDNF, we're able to abolish that effect. And so this is particularly interesting and in line with some other models that show that actually stem cells can modulate disease via indirect mechanisms. If you talk to several Alzheimer's researchers, including me several years ago, we would have said it's impossible for stem cells to actually be effective in Alzheimer's disease, because I think there was this bias that we had that the stem cells had to replace the dying neurons. And now the data are indicating that actually that doesn't have to be the only way that they're acting. They could act via indirect mechanisms, and there's evidence for that in other neurological diseases. So what's next for us? Well, we have a lot of questions that we need to address, not the least of which is if we want to be able to ultimately introduce this into the clinic, we have to evaluate whether or not human cells can perform similar functions in a mouse. And so we have set up a lot of collaborations throughout the world to allow us to do this. And essentially we have 4 large questions that we're seeking to address. We want to be able to identify methods that will allow human cells to enter into the brains of these animals and survive for long periods of time. And there's several issues that we have to consider here. For example, is it important to put sex-matched cells into the brains of these individuals? Do we want to put a female cell into a male brain or vice versa? Likewise, there's an important genetic risk factor for Alzheimer's disease, which is referred to as the ApoE genotype. ApoE is a gene all of us have in this room. It comes in 3 flavors, 2, 3, and 4. I don't know why they didn't start numbering at 1, but the 2, 3, and 4. All of us in this room are either 2-2, 2-3, 2-4, 3-3, 3-4, or 4-4. It turns out the higher the number, the greater your chances are of developing Alzheimer's disease. So in the general population, individuals who harbor a 4 allele only occur about 10% of the time. However, in Alzheimer's disease, they're overrepresented such that they represent anywhere from 40 to 80%. Now that becomes a very significant question, because if you have an individual suffering from Alzheimer's disease who is a 3-3, do you want to go and add 4-4 cells into their brains, which could potentially accelerate the disease process and not ameliorate it? So these are one of the factors that we have to consider. So from this, then we then hope to identify several human stem cell lines that actually can be introduced into the clinic, and then we want to evaluate and compare these human stem cell lines to BDNF mimetics and to other genetically modified stem cells. Perhaps those that might target beta amyloid, for example, might lead to even greater cognitive improvements. And then we set up a collaboration to actually do these studies in monkey brains to see what effect this would have in a higher ordered species. And so here's the team of investigators that we've established. Most of this work will be conducted here at UC Irvine and involves Matt Blurton-Jones, who did a lot of the transplantation studies that I talked about, and Peter Donovan, who's of course the world's expert on human embryonic stem cells. But importantly, we were able to set up collaborations with Richard Boyd at Monash University, who's an expert in tolerance and immunological aspects. Ken Kosick, who works on at UCSB, works on microRNA profiling. Stuart Lipton, who's at the Burnham, who will conduct a lot of the electrophysiological studies. Gene Loring, who's at Scripps, who will also play an important role in molecular profiling of these cells. And Mark Tazinski at UCSD, who's an expert in primate biology. Sorry, the top part got chopped off here, but essentially it shows you the flow diagram of the way we're thinking about doing this. Our first goal here is to establish long-term engraftment of these cells into the mouse brain, and that's going to require a lot of an investigation, a lot of tolerance mechanisms. Then we want to identify a candidate stem cell line and compare it to other different approaches. Such, for example, are the stem cells that we implant better than ones in which we genetically modify, or perhaps better than a virus that happens to be expressing BDNF? And then we have a lot of functional readouts to determine how successful we are in this. Eventually, we hope to identify a lead candidate in primates, and then down the road, enter this into human clinical trials. And I should point out that here at Irvine at the Institute for Brain Aging and Dementia, we are very highly ranked in terms of conducting clinical trials. So we have the experience, I think, to be able to ultimately take this into the clinic. And it might seem strange, although probably not as strange since Rod just went and talked about omentum transplants, of actually doing brain surgery on Alzheimer's patient. And it turns out that our collaborator, Mark Tazinski, actually conducted a somewhat similar trial where he took fibroblasts from patients suffering from Alzheimer's disease and genetically modified them to express nerve growth factor. And these individuals, even though this occurred about 3 or 4 years ago, are doing quite well and actually even show some signs of improvement. So, along the way, we've learned a lot of lessons about transplanting cells and neurostem cells into the brains of these Alzheimer's mice. For example, we know that selection of the appropriate cell type is critical. As I indicated, sex and ApoE genotype probably have to be considered. There might be many others that we have to consider, and that's what the research will have to tell us. We're looking for safe genetic modification of the cells. Questions occur as to what region or regions need to be transplanted. If this is acting through secretion of BDNF, would you be able to just implant the cells into a very restricted brain region, which would obviously offer a lot of benefits? Need to identify a successful immunosuppression regimen, and this is why someone like Richard Boyd is so critical, because it turns out that a lot of the immunosuppression mechanisms themselves directly affect A beta and tau pathology, so some of the simpler ones are out of question right now. We need to identify number of cells to be transplanted, the long-term survival of the graft, and whether or not there's any adverse effects. And lastly, I'll just close with this diagram, since I'm starting to think about these kind of things more and more as director of the Institute, and that is the United States Post Office just issued their first Alzheimer's stamp. And this is available, and it's a pretty somber image of a woman, and you could see that there is a hand here of an individual. This is the caregiver, and the stamp is actually designed to commemorate the caregivers who are really the unsung heroes in this particular disorder. So if you need to buy stamps, I would urge you to buy an Alzheimer's stamp and show some support. And if you can't find it at your local post office, you can go to the postal website and buy it there, and it's nice. So our goal is hopefully to try to delay and prevent Alzheimer's disease. Hopefully that won't be our reward for old aging, and what we hope to achieve is to have everyone live a very active and fulfilling life as they reach their golden years. And these are the people in the lab who did the work. In particular, I want to point out Matt Blurton-Jones, who did the bulk of the transplantation studies here, Masashi, who helped with the microinjections, and Hilda, who did a lot of the work on the behavioral analysis. So, thank you.